Good morning, everybody. First of all, I would like to thank the chair for inviting me and giving me the opportunity to present our work. I also would like to thank you for coming so early. I know that an overdose of hospitality can be sometimes quite hard in the morning, so thanks a lot already. And let's wait for the slides. My talk will be on the integration of ribosome profiling and mass spectrometry. Ribosome profiling, and I'll come to that later. It's a technique which sequences ribosome-protected fragments. Mass spectrometry, I guess you're all aware of that technology, so that doesn't need any explanation. I'm going to talk about two different tool sets that we built. One tool set is actually to use this ribosome profiling information and build extra small open reading frames or open reading frames based on that sequencing information. And then another tool set is really focused towards these small open reading frames, where we have extra metrics based on conservation, based on in silico calculations of ribosome profiling information to gather extra evidence that this open reading frame is actually coding. The first tool is called Proteoformer. It has proteoform in it, and this term is first described, I think, a few years ago. Proteoform is like the combined pool of variability at the protein level. This variability can be based on genetic sequence variations, can be based on RNA editing events, can be based on alternative translation, can be based on alternative splicing. So there's a lot of stuff that makes that at the protein level you have a lot of variability. The second tool is called RiboSorf. It holds ribo because of the ribosome profiling, of course, and sorf because of the small open reading frames. Right now, slides would be quite handy because I could actually show you some data. I tested the slides yesterday, and they appeared quite fine, so that shouldn't be the problem. I also have them on a stick, if necessary. Yes, here we are. So thanks again for coming so early. With this minor delay, we continue. So the first thing, proteoforms doesn't need any more explanation. You all know this already. So there's a lot of variability. Can be introduced on several levels. One level that I didn't mention earlier is the modifications that are going on at the peptide and protein level. So that's something that you actually can measure with mass spectrometry and not with sequencing. So hence, integration of ribosome profiling and sequencing. Okay, this one. You all know Wally? It's sometimes hard to find Wally in the picture. It's also sometimes hard to search for the correct identification of a peptide. The slides are very blurry, I must say. Anyway, Wally is here. Finding Wally, the normal Wally, is sometimes hard already. But it gets even harder if you're looking for Wallies that have an extension, a walking cane, for example, or Wallies that have a truncation, or Wallies that have a single amino acid variant, or very small Wallies. That's why we try to gather extra information from sequencing, in this case, ribosome profiling, to build custom databases in order to find these special wallies. And we use ribosome profiling. And what is ribosome profiling? Well, ribosome profiling, it's a protocol that really captures the small stretches of mRNA which are within the ribosomes while they are translating. So in a way, you more or less have a snapshot of what's going on translation-wise. So you actually measure proteomics, but on the mRNA level. And this information we use to come up with custom databases. There's two types of ribosome profiling. There's the normal type, which would give you this blue signal. The cursor doesn't work either. Which would give you, it's even better now. So the baseline experiment would give you the blue signal. Actually, it gives you a signal all over your coding sequence. And then you have an extra special ribosome profiling experiment which enriches or accumulates ribosomes at the start sites. And this would give you this red signal in the red box, the upper track. And based on that, you actually have the starting point of translation and through what sequence it proceeds. So based on that, you can actually build your custom open reading frames or delineate your open reading frames. And this is what we use to come up with a custom database to search against in mass spectrometry. We built a pipeline to do so. In short, it holds a few steps. First one would be, of course, its sequencing information, so you map the data. And then afterwards, we can extract variation information from that. We can also distill translation start sites from that. Remember the red crisp signal. And then based on that information that we gathered at the RNA level, we move towards the amino acid level and we actually incorporate all these awarenesses and we build a custom database out of that. And this is something we search against using mass spectrometry search engines. The second tool, I also shortly mentioned that in my introduction without slides. It's the RiboSource tool. It has some extra features, and these features are basically similar, so the mapping and all, that's all the same. You also delineate the ORFs, but then afterwards you also measure conservation, and you also measure extra evidence based on ribosome profiling that it's truly being translated, and it's not contaminants or something else. So these are points 4 and 5 to the right. We integrated these two tools in Galaxies so that we are enabling wet lab researchers to actually perform these tools to analyze their own data. And we validated this on two different data sets, a human data set, a mouse data set, where we had matching ribosome profiling information, either public or we generated it ourselves, and mass spectrometry, two types, shotgun and terminal cofradic. Shotgun, all the tryptic peptides, and terminal cofradic, only the N-terminal peptides. And we built custom databases from the ribosome profiling, and then we did the mass spectrometry experiment. This is just a slide to tell you that ribosome profiling and spectral count-based quantitative measures of mass spectrometry correlate quite well, slightly better than RNA-seq and mass spectrometry. And this is the overview result of the shotgun analysis. Left mouse, right human. In gray, you would see what you are able to identify at the protein level by searching SwissProt. The green sections in the pie chart would give you either an improved score or an actual new identification. And these new identifications are basically sequences which are not in SwissProt or novel isoforms or extensions, or to a small extent also upstream open reading frames. This is the overview result set of N-terminal cofradic. In gray, again, is what is annotated in databases, so start sites that we all know. And then the other sections, the red and the yellow, red would give you N-terminal truncated protein forms, and yellow would give you N-terminal extended protein forms. And the smaller pies gives you an idea of stuff that starts either at cognate start sites or at near cognate start sites. So basically, with ribosomal information, you can also pinpoint locations where translation starts at near cognate start sites. Okay. And we were also able to identify stuff that, or genes where you had multiple translation start sites in the same genomic region. These are a few examples. In short, the upper one is peptide identification in N-terminal extended form. The second one would give you a peptide with a single amino acid variation. The third one is actually a peptide cross-spanning a splice junction pointing towards a specific isoform. And then here is a genome overview. You have to the left, in the left bottom, magnification, and you not so clearly can see. There's a track holding the peptides, the N-terminal peptides, which are the light blue bars. So you can see that we are able to identify multiple translation start sites. They start at near cognate start sites. They are all acetylated, and the initiator methionine is also already cleaved off, hence pointing towards really translation start sites. Second part of my talk would be on small open reading frames and putatively coding translation products from these small open reading frames. We all know, or we don't all know, the regular bioactive peptides, which are cleaved from bigger precursors. They all have a signal peptide, so they're directed towards a secretory pathway. In this case, we study micropeptides. These are translated immediately from small open reading frame. So and they are released in cytoplasm because they lack the signal sequence. The last few years, there were some evidence or some manuscript describing, even functionally characterizing, a few of these peptides, micropeptides from small open reading frames. One of the most studied ones are the tarsal-less and in Drosophila and the sarcolipin and myoregulin in human even. Again, this pipeline, just to show you that these extra things happen here. We also calculate conservation on multiple sequence alignment using the phylo-CSF scoring, and we also do the floss scoring. In general, these small open reading frames can occur anywhere. I mean, they can occur in 5 UTRs, exonic, intronic regions, intergenic regions, non-coding regions. And I mean, they can, by chance, they can, I mean, it's easy to detect these. So we have to filter these if you want to get to a more stringent set of sequences. We can filter based on coverage, extra coverage of ribosome protected fragments. We can filter based on translation, which is the floss score that I mentioned, or we can filter on conservation, which is the phylo-CSF that I mentioned, to come up with more stringent sets based on only ribosome profiling information. But in the end, what we also want to do is get some mass spectrometry validation that it's really translated these sequences. There you need extra enrichment, extraction and separation techniques to come up with an enriched sample to only try to analyze most of the time low abundant micropeptides. And there's several options at different levels. I mean, at sample collection, you can optimize at peptide extraction level, at separation level, even at mass spectrometry and validation, you can optimize. What we could also do is bulk search already available data. And we also did that, or are in the middle of doing so. So we use the Pride reshake algorithm to rescan data sets which are available in Pride to really focus towards identifying translation from these small open reading frames. So a few examples, a handful of examples that we already found. This is an example in a long non-coding RNA, a small open reading frame. You see that the conservation is quite well. You have multiple mammalian sequences there. It also has, a pointer would be nice now. It also has nice ribosome profiling coverage. You see that in the lower two tracks. So the upper one is the red signal, pointing towards very good translation start site signal. And the blue track gives you a signal all over the coding sequence of your small open reading frame. Another example also was validated by Pride Reshake already. It's another long non-coding. Thanks. The upper one. No. And then a third one is from the MKKS. It's an upstream open reading frame that we were able to identify with mass spectrometry as well. Also has a very nice conservation and a very good MS validation as well, and ribosome profiling coverage. Attention. While trying to identify these small open reading frames, one must be very cautious. An upstream open reading frame could point to also a 5' extension. A small open reading frame could also point to below threshold identification happening in a reference database. A small open reading frame could also point to some sequence which is available and translated to MBL, or a small open reading frame can also point to variant peptide or within the reference database. So you have to really be careful, and you really have to scan multiple databases to really know what you're talking about. And you also have to take into account some contaminations. Some of these guidelines were described, since it's also a proteogenomics approach, by a very nice review of Nesvitsky quite recently. And we are in the middle of automating these different validation steps here. All this small open reading frame stuff will be presented in a poster on Thursday by one of our PhD students. It holds all the information, all the metrics, all the Pride reshakes, also phenotypic mutations, co-locating with these open reading frames. To conclude, Multi-Omics really helps out to identify novel proteoforms, and we devised 2 different pipelines and a database to actually enable users to make full use of that. And rests me to thank a lot of people from my lab, especially Jeroen, Crappé, Elvis, Ndah, and Volodimir, Olexiouk, and people from collaborating labs from Medical Protein Research, Center for Proteomics in Antwerp, the Compomics group, and also the sequencing facility in Ghent. And I would also like to thank you for your attention.
We'll take 1 or 2 questions.
Thanks, that was very interesting. I'm wondering about the, when you do the proteoformer and build your database, so the ribosome profiling data gives you sort of a general idea of where the translation initiation site is, but presumably you have to look for all the start codons underneath that peak and look at all 3 reading frames and then just infer ORs from there? Is that what Proteoformer is doing?
The main advantage of ribosome profiling is you don't have to go for several reading frames. You actually know where the peak is, so you can distill what reading frame to use. So that gives you a big advantage over RNA sequencing conversion towards amino acid sequences, because you don't actually explode your database.
Okay.
I have a related question.
Yes.
So when you're talking about these near cognate codons,
Yes.
now in the genome and in the RNA, you don't have the conventional ATG, and all the protein databases are driven off of that.
Yes.
So when you are doing proteomic search, then are you basically forcing it and converting those other amino acids into methionines?
Yes.
Or is that the trick? Because all the other people out there, if they don't do that, they're going to miss all of those. So in other words, it might be already out there in existing data sets, but we are under-identifying them, with subject to all of the caveats we have.
I don't have anything to add here. Correct.
Thank you so much.
I have one question.
Yes.
Okay, actually, when you measure those cells, mRNA, or ribosome-based mRNA,
Yes.
what's the size difference? Also very similar, or?
Well, these ribosome-protected fragments are around 30 base pairs.
Yeah, so the overlap to mRNA database overlap is 90% overlap, or just?
mRNA is the complete mRNA. Ribosome profiling is the coding sequence. So all the UTRs are
We're talking about the
discarded.
mRNA. mRNA should be similar to
mRNA, which is actually being translated. So we're talking about the coding sequence if we measure with ribosome profiling. So all the UTRs are gone, let's say, which is quite a part of your mRNA in some cases.
Thank you.
You're welcome.